Welcome back, students. Having done the structure of DNA, let us now move on to packaging of DNA. Where would it be packaged? Of course, within the nucleus in case of eukaryotic cell and in the nucleoid region in case of prokaryotic cell. So we'll do this packaging topic in two videos. First, I'll be taking up prokaryotic cell, DNA compaction or packaging in prokaryotic cell, and a little bit of briefing about the eukaryotic cell as well with some calculations, but the details we will take up in the next video of eukaryotic cell. So coming to DNA compaction in prokaryotes. You know what are prokaryotes? Primitive nucleus, pro meaning primitive, carry-on related to nucleus. So they have a very poorly developed nucleus, primitive nucleus, not well defined. The boundaries are not there, that is the nuclear membranes are not there. So chromosomes or the genetic material is not bound by nuclear membrane in prokaryotes. But still, this is a bacterial cell. The chromosome is not bound, but it is not scattered anywhere. It is not scattered throughout the cytoplasm. It is held or concentrated at a particular region called nucleoid. Nucleoid, devoid of nuclear membrane. An area devoid of nuclear membrane. So how is it concentrated there? We'll go through the steps of packaging or compaction. How is it compacted? The circular DNA of a bacteria, it has to be made into a compact form to fit into a bacterial cell. Why? Because if I take the example of E. coli, a rod-shaped bacterial cell, its dimensions, if you see, length would be around 2 to 4 microns and radius would be 0.5 microns. So when you see the size of the cell, which comes to approximately 1 micron width, the cell size, you compare it with the nuclear material, that is the genetic material, it comes to 1.6. The length of the DNA is around 1 millimeter or so. So here I have given E. coli I have taken as 1.6 mm. So it's quite long, around 1000 times larger, 1000 folds larger than the size of the cell. So how would such a big material fit into the cell, which is smaller than it, much smaller than it? So that compaction process we'll go through. It comes through formation of loops. The circular structure has to loop out. How does it form such loops? So that it can be held in a place by certain proteins which are called non-histone proteins. We'll talk about the term histone in detail when we come to eukaryotic compaction of DNA. Here it is non-histone proteins, that is DNA binding proteins. So these yellow colored portions are all those proteins. And the circular DNA, see how it is coiling like looping. This will gradually come inward. You can see here. So these are the loops being formed of the DNA. So the the non-histone proteins compact them by pulling them inward to form such loops. We call those areas or that kind of looping as loop domains. So when such loops are formed, you can see the non-histone proteins holding the loops together. There are many other factors holding them together, but I am limiting the knowledge to 12th class students. So these non-histone proteins help in the formation of such loops and they hold them in place together. But then this itself is not small enough now. It has to be further coiled. So the further coiling, we call it supercoiling. How is further compaction accomplished? It is accomplished through a process called supercoiling. That means coiling back on itself. And this is controlled by enzymes called gyrase and topoisomerase. These two enzymes, like here, these are the enzymes which are holding them, which are pinning them in place. So these enzymes cause further coiling, which is called supercoiling. Can you see this? You take a rubber band, a long rubber band in your palms, in between your palms, and just rub them. How would they twist and turn over each other? So that kind of supercoiling occurs because of these enzymes which further concentrate them and hold them in a particular region within the cell, and that is called the nucleoid. So main thing about the compaction is DNA looping, which is done by these non-histone proteins or DNA binding proteins, and further supercoiling by the enzymes gyrase and topoisomerase. These two make the chromosome compact enough to be fit within the region called nucleoid. So that's it about the prokaryotic cells. The question to that mainly appears in the board is how is DNA organized in the nucleoid region? It is by the formation of large loops held by proteins that should be mentioned in the nucleoid region. Of course, supercoiling is also there, which is also one of the ways in which how it is concentrated. So both looping and supercoiling. Coming to a calculation based on this. Suppose you are given a length of E. coli DNA as 1.36 mm. This is a question given in your NCERT in between the lesson. Can you calculate the base pairs? Of course, you know we have learned through Watson and Crick model the structure of DNA and all that distance between two base pairs is around 0.34 nanometers, right? So let us convert it into meter. 1 nanometer is 10 to the power minus 9 meters. So this is how you convert it. Then length is given, length of the DNA is given in millimeter, and this is millimeter, okay. So you convert the millimeter into meter, so it comes to 1.36 into 10 raised to the power minus 3 meters. So we have both these in meters now. Let us consider what we have to calculate, that is the base pair as Y. This is the formula that you have to apply. Length of the double helix DNA is calculated by total number of base pairs into distance between the two base pairs, any two base pairs. So total number of base pairs is total number of base pairs we have to calculate. Length of the DNA double helix is given and distance between two base pairs we know 0.34 nanometer, which is converted into meter here. Okay, so you calculate that, you get 4 into, you divide these two numbers, you will get 4. 10 raised to the power minus 3, you take this minus 9 up, becomes plus 9. So minus 3 plus 9 is 6. That is the number of base pairs. I hope that is clear about the prokaryotic DNA. Now coming to the size of a DNA helix in a single cell in human. When we come to packaging of DNA in eukaryotes, we first have to understand the size of the DNA in a human cell. You know, human genome, we generally take haploid cell to count the genome. It is approximately 3 billion base pairs that is packaged into 23 chromosomes, haploid. So double it for diploid cell, 23 pairs of chromosomes. How many base pairs? It will be 6 billion base pairs. So total base pairs in a diploid cell is 6.6 into 10 raised to the power 9 base pairs. Distance between two base pairs we already know. We have done this in the previous frame, convert it into meter. Now, length of the DNA double helix, the same formula, total base pairs into distance between the two. So you can easily calculate the length of the DNA because you have the total number of base pairs, that is the genome size as 6.6 into 10 raised to the power 9, and you have the distance between two base pairs, consecutive base pairs, that is this, multiply this, you get around 2.2 meters, around 2.2 meters, a little more. So that is the size of the DNA in one cell, one diploid cell. Now, what is the size or dimension of a typical nucleus? Naturally, our cells are microscopic, so nucleus is even more smaller. So it's somewhat 1 micron, that is 10 raised to the power minus 6 meter, comes to around 1 micron. It's very small. How does such a long 2 meter DNA fit into a 1 micron nucleus? This is what we have to study. Okay, coming to that, the length of the DNA double helix in human cell comes to 2.2 meters or so. So the length which is far greater than the size of the nucleus which holds it. And if all the DNA in a single human cell was linked end to end and it is stretched out, that is that 2.2 meters when you stretch out, that is it, 2.2 meters comes to around 6 feet long. This is just a knowledge thing I have written. 2.2 meters is around 6 feet long approximately. This is about one cell, one human cell. Okay. Now you think just estimate how many cells we have in an adult human. Around 50 trillion cells approximately. Every cell has got that 2-meter DNA, right? So you calculate the total DNA in our body. It must be around 100 trillion meters. 50 trillion cells into 2 meter per cell. 100 trillion meters. Just imagine, so amazing. Such a huge mass of DNA, length of DNA we have, and that is what people relate it to in layman's language. They make you understand like you can stretch it out to the sun and come back more than 300 times or so, something like that. Okay, just wonderful. So if the length, now another question based on it, if the length of a double helix DNA is 1.7 meters, same type of calculation, find the number of base pairs, same thing, distance between base pairs, length is given. Only this you have to convert into meter in this case, in case of a eukaryotic cell. So consider the base pairs number as y. So same calculation. So number of base pairs is length of the DNA divided by distance between two. Length is given 1.7 meter, distance between two base pairs you have converted into meters. 1.7 upon 0.34 you will get 5 into 10 raised to the power 9 base pairs. That's it. Okay, now finally, how is such a long 6 feet long DNA or 2.2 meters long DNA fit into the nucleus, which is very small? How is that compaction done? Okay, we are going to read that in detail in the next frame, but I'll just tell you the unit of that whole process of how it is compacted, how this chromatin is formed, how it is. So the DNA, the long DNA strands, how are they compacted into structures called chromatin? So the unit is called nucleosome. The basic unit of a chromatin is called nucleosome. It is DNA plus histone. You can see this ribbon-like structure is the DNA shown, somewhat less than 2 turns it takes around. Proteins called histone proteins, which are positively charged. These proteins are 4 in number into 2. 4 types of histone proteins into 2. So total 8. We call it as histone octamer. The names are H2B, H2A, H2B, H3 and H4 into 2. Each one into 2. So down there is one, same H3, H3, H2A, H2A, H2B, down H2B, H4 and H4, like that. This makes a octamer called histone octamer, and these are positively charged proteins. DNA being negatively charged, they pack them so tightly, and this is one extra H1 linker histone protein which seals the open ends. The entry end and the exit end is sealed by H1, making it very compact. So the whole DNA is having such kind of compaction, and then you call it chromatin. In details, we will talk about what are histone proteins, how DNA is compacted, how it fits into the nucleus, all this in the video that continues this one. Okay, so I hope this much is very clear. Happy reading. Keep watching. Thank you.